Hello friends, I am Dr. Marathe from the Department of Microbiology. Today I will discuss about a modified form of TCA cycle that is called as Glyoxylate cycle. Glyoxylate cycle. Okay. Why this is known as glyoxylate cycle? The answer to this question is what? In this cycle, one of the unique intermediate is there. That intermediate is called as glyoxylate, okay, or glyoxylic acid. Now, why this modified form of TCA cycle is there? What is the necessity or what is the need? What is the need of this bypass mechanism of TCA cycle? See, especially when we talk about the TCA cycle, acetyl-CoA, which is a 2-carbon containing compound, when it enters in the TCA cycle, okay, the 2-carbon containing compound condenses with the oxaloacetate, it forms citrate. From the citrate through the intermediate cis-aconitate, it is isomerized to isocitrate. From isocitrate, alpha-ketobutyrate to succinyl-CoA to succinate, fumarate, malate, and again oxaloacetate is formed. But during this normal pathway, what happens? Whatever the 2-carbon fragment of acetyl-CoA, it was incorporated into the TCA cycle, it gets lost during the two reactions. That is conversion of isocitrate. If you remember my first TCA cycle video, you will come to know through the isocitrate. When the isocitrate is converted to alpha-ketobutyrate, there one carbon dioxide is involved, okay, CO2 is involved, okay, that means there is one loss of carbon atom. Second, alpha-ketobutyrate to succinyl-CoA, another CO2 is involved, that is another loss of carbon, means 2 carbon atoms, they are removed through the cycle, through these 2 steps where the carbon dioxide is involved. Means whatever the 2-carbon containing compound, that is acetyl-CoA, when these 2 carbons were incorporated into the TCA cycle, they were lost and remaining 4 carbons, these are recycled. Okay, but this bypass mechanism is essential for certain algae and certain plants. Generally, especially this pathway, that is glyoxylate cycle, it is operated into the particular organelle of germinating seeds, okay, in case of plants. So those organelle, they are known as glyoxysomes. So glyoxysome is a particular organelle where this cycle is operated. It's called as a glyoxysome, especially in a germinating seed. In a germinating seed, they require organic compounds, carbon-containing compounds. Okay. So what happens during the reaction? How this bypass mechanism occurs? Then I will try to explain. Okay, so acetyl-CoA along with oxaloacetate, which is a 4-carbon containing compound, it contains 2 carbon, it forms 6-carbon containing compound, and that 6-carbon containing compound is called as citrate. Okay, so this is a tricarboxylic acid compound. Okay, by the enzyme citrate synthase, during the reaction, coenzyme A, it is removed. Acetyl-CoA contains a coenzyme A, it is removed. Okay, so the 2-carbon fragment of this acetyl-CoA, it condenses with the 4-carbon containing compound oxaloacetate, and it forms what? 6-carbon structure called citrate. Okay, now this citrate, through the intermediate like cis-aconitate, it is converted to isocitrate by the enzyme aconitase. Okay, in the first reaction there is addition of water, there is a removal of water molecule, and in the subsequent reaction there is a loss of water molecule. So whatever the structure it is formed, it is the isomer of citrate, and that isomer, it has a structure like this: COOH, CH2, CHCOOH, COH, H, 1, 2, 3, 4, 5, and last is what? CO2. This is called as isocitrate. This is known as isocitrate. So isocitrate is the intermediate of TCA cycle. It is the isomer of citrate which is formed from the acetyl-CoA and oxaloacetate. Okay. Now the next reaction which generally happens in the TCA cycle is conversion of acetyl-CoA into the alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. But this reaction is bypassed. So ultimately the loss of carbon in the form of carbon dioxide is stopped. Okay. So instead this isocitrate, it is lysed. Okay. Now this bond is broken down. Okay. So this 6 carbon containing compound, it is lysed, 4 carbon structure and a 2 carbon structure it is formed. Okay. So that 4 carbon structure is what? COOH. This one is COOH, then CH2, then CH, and this H is transferred from this carbon to this carbon, CH2 and COOH. Now this is a well-known component of TCA cycle. This is known as succinate. Succinic acid. Okay. Yeah. But another structure that is formed is called as CH double bond O, that is COOH. Now this structure, this is the unique form. This unique structure is called as glyoxylate or glyoxylic acid. Okay, so this glyoxylic acid, it is a unique component of this bypass mechanism. That's why this cycle is known as glyoxylate cycle. Okay, so this glyoxylate form, it reacts with another molecule of acetyl-CoA plus, I am writing here, plus CH3 C double bond O S COA. So this glyoxylate, okay, this glyoxylate reacts with another molecule of acetyl-CoA. Okay, now actually what I said, whatever the succinate is formed, that succinate is converted to fumarate. Fumarate is converted to malate. And this malate in a regular process is converted to what? Oxaloacetate. Now what happens when the glyoxylate, it reacts with the acetyl-CoA. Okay, so when it reacts with the acetyl-CoA, it forms this glyoxylate and this acetyl-CoA. Okay, that actually it is through the enzyme malate synthase, and here the enzyme is isocitrate lyase. So through the enzyme isocitrate lyase, this reaction is catalyzed by the enzyme isocitrate lyase. Isocitrate, it is splitted into 2 compounds. One is known as a 2 carbon compound, glyoxylic acid or glyoxylate. Another is known as a 4 carbon containing compound, which is the intermediate of regular TCA cycle, that is succinate. Through the normal TCA cycle, this succinate is converted to fumarate to malate. But another malate molecule is formed. How? By reaction of acetyl-CoA and glyoxylate. So this acetyl-CoA and glyoxylate through the removal of COASH. Okay, this reaction is accompanied by hydrolysis. H2O is split into H plus and OH minus, and this structure is formed COASH and malate. Okay, so this malate, all of you know the structure of malate. So this malate COOH, CH2, CHOH and COH. This is malate. Okay, so through this 1, 2, 3, 4 carbon containing compound. So 1, 2, 3, 4 carbon group. This malate is formed. this malate. So what is the outcome of this process? Outcome is nothing but what? This is the bypass mechanism of TCA cycle. Some steps are avoided through this cycle. Those steps are first and second step, means conversion of isocitrate to alpha ketoglutarate and alpha ketoglutarate to succinate CoA, where two carbon dioxide molecules are evolved. That loss of carbon is avoided and is maintained. So whatever the malate is formed, that malate goes to, it is converted to oxaloacetate. So the oxaloacetate it is directly converted into enzyme kinase into PEP, phosphoenol pyruvate, PEP, and by the process of reversal of glycolysis, that is called as gluconeogenesis. Through the process of gluconeogenesis, that PEP through certain bypass mechanisms, it is converted to glucose. That is nothing but gluconeogenesis. So carbon organic compound is synthesized. Here the main purpose is what? The synthesis of organic compound, and that is glucose. Okay, not removal of CO2. So the removal of CO2 is avoided through this cycle by the formation by the bypass mechanism, which is catalyzed by two enzymes. One is isocitrate lyase, which is responsible for lysis of isocitrate into glyoxylate and succinate, and another enzyme which is responsible for reaction between acetyl CoA and glyoxylate, that is malate synthase, which is responsible for synthesis of malate. That malate can be converted to oxaloacetate. Oxaloacetate can be converted into direct phosphoenol pyruvate, and through the reversal of glycolysis, that is called as a gluconeogenesis, it will be converted to glucose. There will be formation of glucose, which is an organic compound, carbon containing compound, which is essential for the germinating seeds. Okay, so this is the bypass mechanism, and that's why due to the presence of one unique intermediate in this cycle, it is labeled as glyoxylate cycle. Thank you.